Hello students, our today's topic is cytokinin signaling in plants. Cytokinins are plant hormones. They are a class of molecules that are unsubstituted adenine derivatives such as isopentenyl adenine and trans and cis zeatin, which are common in plants. Phytohormones are well-established regulators that are involved in nearly every aspect of plant life, including plant growth and development and the response of plants to changes in their abiotic and biotic environment. Actually, phytohormone biology can be divided into three parts. First is metabolism, means biosynthesis and degradation. Second is transport, and third is perception and signaling, right? Signaling Cytokinin signaling process is a crucial part of almost all stages of plant life, from embryo patterning to apical meristem regulation, organ development, and eventually senescence. The cytokinin signaling pathway involves the coordination of three types of proteins. First is histidine kinase receptors to perceive the signal. Second is histidine phosphotransfer proteins to relay the signal. And third, response regulators to provide signal output. Although much is known about how this cytokinin signal is perceived and initially regulated, there are still many avenues that need to be explored before the role of cytokinin in the control of plant process is fully understood. We'll start with the cytokinin signaling components. So first is histidine kinase receptors. Histidine kinase receptors to perceive signal, right? Three types of histidine kinase receptors are there in cytokinin signaling. First is Arabidopsis histidine kinase 2, AHK2. Second is Arabidopsis histidine kinase 3, AHK3. And third is Arabidopsis histidine kinase 4, that is AHK4. AHK4 is also known as cytokinin response 1, CRE1, and wooden leg, abbreviated as WOL. Actually, first cytokinin receptor gene was described as wooden leg, WOL gene, in Arabidopsis, which later became known as cytokinin response 1, CRE1, and now Arabidopsis histidine kinase 4, right? The Arabidopsis cytokinin receptors kinases contain three parts. Just look at this diagram. First is a conserved extracellular cytokinin-binding domain called the chase, this green. Chase cyclases histidine kinases associated sensory extracellular domain. Second is a histidine kinase domain, this blue in color. And third is a receiver domain, this yellow in color, right? So, three parts of histidine kinase receptors that perceive the signal, right? Next type of protein in the signaling chain are histidine phosphotransfer proteins, right? Histidine phosphotransfer proteins is to relay the signal, right? Just look at this diagram, this red in color. Histidine phosphotransfer proteins, AHPs, is to relay the signal, right? There are 5 Arabidopsis histidine phosphotransfer proteins are there, AHPs. In Arabidopsis, the pseudo-HPT AHP6 functions as a negative regulator in cytokinin signaling pathway. However, a recent study of canonical and pseudo-HPTs from rice showed that pseudo-HPTs can also play a positive role in cytokinin signaling and thus function differently than those from Arabidopsis, right? Third type is response regulators, right? These are response regulators. Two types of response regulators are there, ARR and BRR. The 23 ARR genes fall into two main groups on the basis of their sequence similarities domain structure, and transcriptional response to cytokinin. The type A ARRs, right, are comprised of a receiver domain and a short carboxyl terminus, and their transcription is rapidly elevated in response to exogenous cytokinin, right? So these are considered to be the primary response genes. Then the type BRRs, in addition to the receiver domain, this yellow in color, have a carboxy terminal output domain, right? That has a DNA binding GRAP domain, this green color, and a transcriptional activation domain, right? So this part is response regulator. Next is cytokinin signaling. There are two types of cytokinin signaling has been identified. The first is two-component signaling and the second is multi-step phosphorylase signaling. First, we'll start with the two-component signaling, right? Two-component signaling, simply two components, right? This one component, that is chase domain and kinase domain, the receptor kinase, right, to perceive the signal, right? And second part is response regulator, right? So simple two-component signaling systems are comprised of a receptor kinase and a response regulator, right? Kinase autophosphorylates in response to an environmental stimulus, right? From here, any environmental stimulus will cause this kinase to autophosphorylate, right? And this is a phosphoryl group. This phosphoryl group is then subsequently transferred to a conserved aspartate residue within the receiver domain of response regulator, right? In multi-step phosphorylase signaling, here multi-step phosphorylase system are comprised of sensor kinase receptors, right, to perceive signal, that have both kinase and a receiver domain, chase domain, kinase domain, and receiver domain, right? Then second, histidine phosphotransfer proteins to relay the signal, here this red in color, and third is response regulator, right, to means output signal for output signals, right? So all the three are involved here in multi-step phosphorylase signaling, right? Ligand binding first alters the autophosphorylation activity of sensor kinase, right? Then phosphoryl group is transferred from the autophosphorylated histidine to an aspartate residue within the fused receiver domain. This is the receiver domain, and then to histidine residue in a histidine phosphotransfer protein here, and ultimately to an aspartate residue in the response regulator, right? I will also discuss a general model of cytokinin signaling in Arabidopsis that has been described by Romanov et al. 2018. Here, cytokinin is perceived by chase domain, that is, cyclase histidine kinase associated sensory extracellular domain, this green in color, right? So cytokinin is here, it is perceived by chase domain, right? As chase domain containing hybrid histidine kinase receptor, CHKs, right? Either at plasma membrane or at endoplasmic reticulum membrane. Now listen, subcellular sites for the onset of cytokinin signaling are still uncertain and experimental data are in part controversial, right? If you want more information on this topic, please read a review paper by Romanev et al. 2018 titled cytokinin signaling from the endoplasmic reticulum, that is from the ER or from the PM? That is the question, right? Now, after intramolecular phosphorylation, the signal is transferred to HTP proteins here, right? AHPs, which means shuttle between the cytokinin and the nucleus, where the phosphorylate response regulators are there, right? All the regulators are in nucleus, right? There are two different type of response regulators. Just look at this diagram. Here, this is nucleus, plasma membrane, cytoplasm, and nucleus, right? So, response regulators are present in the nucleus. And this chase domain, this is on the plasma membrane, right? So, two different kinds of response regulators involved in cytokinin signaling in plants. First is type B response regulators, RRB, right? Which are MYB transcription factors and activate the transcription of a group of target genes, and type A response regulators, ARR. The RRAs negatively regulate the signaling pathway, most likely by competing with the RRB for phosphorylation signal from the HPTs, right? So this is all about cytokinin signaling. If you have any question, please write in the comment section so that I can answer, right? Thanks for watching. If you like this video, please make sure to subscribe for more. Thank you.